Vicki Levasseur', and I spent the summer in Houston, Texas doing research at Baylor College of Medicine. Baylor College of Medicine is located in the Texas Medical Center, which is a very large area full of medical-related buildings, and within Baylor itself there's about 90 research and patient care centers, and the funding for research there's a lot of funding available, and I participated in a research program that was centered at allowing undergraduate students to be able to spend time in a professional lab, and it was called the SMART program. So the SMART program, not only did I conduct research, I also attended daily lectures presented by professional scientists who shared what kind of research they were doing to us, and it was a nice thing to do to take a break from the research that I did. I learned several different laboratory techniques while I was in the lab I worked in, Western blotting, which I'll explain later, and then several other techniques. And something special I'd never done before was work closely with mice and rats, doing hormone treatments, brain tissue dissection, and I learned how to do a couple of surgeries too. So my main project was focused around post-traumatic stress disorder, which is an anxiety disorder that results from exposure to some sort of traumatic event, such as a war experience or maybe a car crash, something like that. And the symptoms that a person with PTSD exhibits are avoidance of things related to the event that they experienced, a loss of interest. Sometimes they have flashbacks, they feel tense or on edge at times, they have trouble sleeping, and maybe they have outbursts of anger. So I studied something related to P11, which is a protein located in the brain. And when it's activated, it influences the release of serotonin, and this is a neurotransmitter that kind of affects mood. And so we wanted to see if P11 levels in the brain were associated with post-traumatic stress disorder related symptoms. So there's several regions in the brain that deal with stress and mood. So the prefrontal, this is a rat brain by the way, so the prefrontal cortex is an area that deals kind of with cognitive behavior and decision making, and then the hypothalamus deals with kind of primitive things about the human or rat, such as temperature regulation, hunger, thirst, and sleep and wake cycle. And then the hippocampus is an area that deals with like memory formation and space, the way you kind of feel yourself in space. And also the amygdala is an area that deals with forming memories and storing memories, and also specifically memories of like historical events that have occurred or emotional events. And then the cingulate is also an emotion forming and processing area in the brain. So to see how P11 was being expressed in the rat brain tissues, we had to dissect from these areas in the rat brain. So we isolated these areas, and then we took the tissue and kind of homogenized it, which means just broke it up so that it was in smaller, more like uniform, like liquid-like texture. And then we wanted to see how much protein was in these samples of tissue so that we could use a Western blot, which would tell us how much protein was in each of the samples of brain tissue, how much P11 protein. So we did that with something called gel electrophoresis. So we took the samples of brain tissue and put them into these plastic-like things you can see here, and we injected the samples, and then the samples would run from negative to a positive charge based on their molecular weight or their general size. So after that, we transferred the proteins onto this thing called a PVDF membrane, which just takes the proteins from the original gel and then puts it onto the membrane. And then we applied an antibody that detects P11, so you can visualize how much is on the membrane. And then we probed it with another antibody that attaches to the first antibody so that we can see how much P11 we have. And this is kind of what the results for a Western blot look like. Down here is the important part, because we knew that P11 has a molecular weight of about like 11 kilodaltons, so we were seeing a lot of it here in the samples of the amygdala, the cortex, and the hippocampus. So that was pretty cool. And we didn't get a lot of Western blotting done because it took a long time to treat the rats with the proper hormones and dissect them and then do all the Western blotting. So the results we got here were very preliminary, but they showed us that P11 is present in the areas associated with post-traumatic stress disorder. So that's kind of what I learned, that P11 is something that the people in my lab could study further to see if maybe it could be used as, it could be blocked. The transmission of P11 could help maybe block the symptoms of post-traumatic stress disorder. And then also, in more general terms, I feel like I made a positive contribution to this research study, and I learned how to quickly acquire new skills. And personally, I improved my communication skills with the people in my lab, as well as when I was presenting my research to other students at the end of the program. And I also learned to work effectively with other students who were at a higher level of education. There were a few graduate students in the lab, and also with research technicians and other professional scientists. And academically, I'm more confident in the lab setting, and this will help me in my future courses at Cornell, as well as in the future when I attend graduate school or medical school. And so professionally, I'm encouraged to become a medical doctor and or conduct neurologically related research. So I would like to thank the donor for this fellowship, George Sharp, my faculty sponsor, Craig Tepper, and the site supervisors, the head of the SMART program, Dr. Slaughter, the head of my lab, Dr. Shaila Hamani, and the research technician I worked with, Andrea Reyna, and then the Cornell College Fellowship Program.